la scienza. Nella città ai confini, dove le barriere, se esistono, vengono soffiate via. Qui c'è un'energia unica che cambia il mondo, genera intesi, supera i limiti. Qui sono nate aziende che hanno colto idee e opportunità. Qui le menti brillano, pensano più forti, creano con una visione chiara. Qui, dove l'innovazione è al servizio dell'umanità, il futuro avrà una casa. Qui, dove inizia il futuro.
Sono qui per presentarvi la nuova edizione di Trieste Next. Il titolo della 14ª edizione di Trieste Next è La vita dentro, dialoghi tra scienze e tecnologie. 3 giorni per riflettere su come la scienza e la tecnologia ridefiniscono la vita, su come le discipline STEM dialogano con le scienze umane e su quali nuove domande emergono per il nostro futuro. Medicina di precisione, intelligenza artificiale, sostenibilità ambientale, robotica, biotecnologie e tecnologie quantistiche sono solo alcuni dei temi al centro di Trieste Next. È questo il fil rouge del festival, con un programma di oltre 100 eventi in inglese e italiano e oltre 300 relatori di fama internazionale. E ancora, il premio Trieste Next Science Book of the Year, le 70 attività per le scuole e i 40 spazi espositivi e laboratori aperti a tutti in Piazza Unità. Prima di accogliere gli ospiti del prossimo evento, a nome dei promotori e copromotori della manifestazione, vorrei ringraziare tutte le istituzioni scientifiche di Trieste, città della conoscenza, e le istituzioni che hanno collaborato con noi, il main partner del festival, i partner e i content partner della manifestazione per il contributo fondamentale. Grazie anche ai media partner e ai partner tecnici. Infine, un grazie speciale ai 200 volontari di questa edizione. Ricordatevi anche di controllare il programma aggiornato degli eventi su www.triestenext.it, al quale potete accedere anche da questo codice QR. Buon ascolto e buon festival!
So, good morning everybody, and thank you for being here today. It is my pleasure to talk here at this fabulous meeting. I'm Michela Milani, and I am a project leader at the San Raffaele Teleton Institute for Gene Therapy. And today we are going to talk about new perspective on in vivo gene therapy. So this is my email. If you want to contact me after the conference, and if you have any doubt or question or curiosity, please just send me an email. So this is the outline of the presentation, so what we are going to talk about today, so you know how long it's going to be before the end of the presentation throughout the slides. So the first point that we are going to talk about is that mistakes make us what we are. Then we are going to talk about the concept of gene therapy, the wish list of a gene therapist, the difficult relationship between hemophilia and HIV, and in the end, we're going to talk about new avenues for in vivo gene therapy. So let's start from the beginning. And so mistakes make us what we are. I think that this is a concept that resonates with most of us. So throughout our life in general, there are trials and errors. And through these errors is actually what shape us as an adult person and how we actually grow throughout our life. And actually, this is not very different from what happened from this very tiny blue organism. I don't think it's going, probably in the past it was blue, but I found this picture and it was very nice, so I decided to take it anyway. And actually, mistakes is what, throughout the millennia, brought this little worm in the primordial growth to become what we are, so a very complex organism. And it took around 4 billion years of mistakes that allowed us to evolve from a very simple unicellular protozoan to a complex organism that we are now today. There is a price we pay for these mistakes that actually drove all the entire evolution of our species and all the other species that are in the world. So let's talk about some numbers. So in our organism, there are about 100,000 billion cells, quite a lot. And each cell contains 2 copies of what we can think about is an instruction manual of around 3.2 billion letters. And we call this instruction manual the genome of the cell. So it's DNA that actually encodes all the function that the cells needs to take throughout its life. But the question is, how many letters are 3.2 billions? So I got curious about this, and so I tried to do some calculation about how many actually letters are 3.2 billions, because if you think about it, it's quite a big number, no? So I don't know how many of you knows what is this encyclopedia, because I guess most of you are too young to know it. So this is the La Piccola Treccani, which is a reduced version of the old-style Wikipedia that we had before internet. Essentially, it is composed of 12 volumes, and of course, each one of these volumes contain all the knowledge of humans, let's say, condensed in these 12 books. And I thought, okay, maybe 3.2 billion letters are inside an encyclopedia. But actually, after doing some calculation, and if you're wondering, yes, I counted how many letters are on a page, actually I found out that to reach 3.2 billion letters, you need 25 of these. So let's go back to the numbers. So we have 100,000 billion cells, and inside each one of these cells, there are 2 copies of 3.2 billion letters instruction manual, which corresponds to 25 Piccola Treccani. So quite a huge number of letters. So each time that a cell divides, okay, it needs to copy all this entire instruction manual to give a copy to the 2 daughter cells that results after the cell division. The problem is, if you think about manually copying 3.2 billion letters, known that this corresponds to 25 times this encyclopedia, you can understand that it's quite easy to insert some errors. And the problem is that some errors are minor and do not have any consequence on cell functionality. So it means that even if we have an error, the cell is still able to read what is written inside this instruction manual. On the other hand, some other errors may disrupt the meaning of the sentence, or changing it. And so this is what leads to genetic diseases and tumors in our life. And this is the price we pay for the evolution that mistakes allowed throughout these 4 billion years. So what is gene therapy? Now that we know what is a genome, what is the instruction manual that is inside the cells, and what is a genetic disease. So it's a disease that arises from one single error, usually inside this huge instruction manual that is inside every cell. So gene therapy is, I like to call it the promise of a cure for genetic diseases. And it's based on the concept of using genes to restore a missing function in the target cell. So in this case, the drug that we want to deliver to the body is the gene. So if we go back to our previous example of the genetic material as an instruction manual, it means that we want to put back the right sentence inside the genome of the cell in order to make it again possible for the cell to read the sentence that it's not able to read anymore because of the mistake. So this is gene therapy. But how can we deliver a gene to a target cell, so to the cell that is suffering from this mistake? So actually what we use in the lab are, and not only in the lab now, also in hospitals, because gene therapy is now a real drug that can be administered to people with the genetic disease. So we use viruses. And why do we use viruses? Because they are, let's say, the perfect vehicle to deliver our genetic material, so our sentence, correct sentence, inside the cell. So, and essentially what we do in the lab is turning foes into friends. And I like to start from this old Danish proverb that is, there is nothing so bad that is not good for something. So we are used to think about viruses as something that we must be, let's say, afraid of, especially after the pandemic, I have to say. And, but what are viruses? So viruses are very different from bacteria, because bacteria don't need us to survive. They just stay around in the environment and they survive by themselves. They just don't care if we are there or not. So they're going to survive after many years after the human beings are not going to be on this planet anymore. On the other hand, viruses are specialized obligate parasites. So this means that they need the host to be infected to survive. So they cannot replicate without a person that is infected or a host, an animal, whatever. And once they enter into a cell, they are able to turn the cell into a virus factory. And how do they do that? They have inside them, so if we look at this virus, inside this, let's say, protein-based core, there is another instruction manual. And this instruction manual is actually contains the instruction to build other viruses. So what happens when a virus infects a cell is that it transmits its own instruction manual to the cell, and then the cell starts reading the instruction manual of the virus and produce other viruses. So this is what happens every time that you got infected by a virus. And so they are super efficient in delivering instruction manuals to the cell, so genetic materials to the cell. So this is why they are the perfect vehicle for us. So what do? We engineer viruses in the lab, and we remove their own instruction manuals, so the instruction to make other viruses, and we replace their instruction manuals with the sentences that we need to correct the errors that are in the instruction manuals of the affected cell, in the case of the genetic disease. So they are essentially the perfect Trojan horse, so to deliver something that we need to go inside the cell in a way that the cell is not aware of. And so we can divide gene therapy in two different strategies. Okay, so today we're going to talk mainly about in vivo gene therapy, so what is on the right of the slide, but it's not the only possible strategy, because we talked about how we can deliver a gene, so the right sentence inside the cell, but how can we reach the cell? Because now we have an engineered virus, but this virus, which we call viral vector, needs to enter into the right cell to get the cure possible of the genetic disease. So we have two strategies. So one possibility on the left of the slide is to collect the cell from the patient. So we have affected cell, for example, in the hematopoietic system, so blood stem cells. So this is quite easy to do. So we can collect these cells from the bone marrow of this patient. We can put them in culture in the lab, and we can engineer them. We put them in contact with our viral vector that goes inside the cell and bring inside the cell the correct sentence. And then, once the cells are corrected, we can transplant them back into the patient. So this is what we refer to as an autologous transplant. So this is exactly what we do when we do donor-compatible transplant, for example, in leukemia, or other type of tumor. So we have a donor, we collect the stem cells, and then we transplant them into the patient. So here we do the same thing, but we collect the same cells of the patient. We correct them, and then we infuse them back into the patient with an autologous transplant. Is this possible for every genetic disease? Maybe not. Why? Because if we think about that we have many organs in our organism, let's say that we want to correct some cells that are in the brain. Can we take out the brain for a person, correct it in vitro, and then so in the lab, and then put it back? Of course not, we can. Can we do this for the kidney, the liver, all the main organs, solid organs that we have in our organism? No. Now we are not able today of doing this. So we develop an additional strategy, which is referred to as in vivo gene therapy. So in this case, what we do, we directly infuse our viral vectors inside the body. So this can be done through the circulation, for example. This can be done directly in the brain. This can be done on the skin, like with the gel, for example. And so we can deliver our virus directly to the organs that we need it to go. So what is the wish list of a gene therapist? So today, actually, gene therapist is not yet a medical specialization, but I guess that in the future it can become a new, so you call your doctor and you say, oh, I need a gene therapist, but not for now, but maybe one day you can. And so what is the wish list of a gene therapist? So there are a few things that we need to take into consideration. Safety first. So actually this is true for gene therapy, but it's true for every drug. So whenever you do a clinical trial, the first aim of the clinical trial is to show that your drug is safe, not that it's efficacious. So the first thing is safety. And what is the problem? Is that our organism judges the book from their cover. Maybe sometimes we do it as well. But the problem with our organism is that we have a very efficient immune system. And here we are proposing to infuse inside the circulation something that looks like a virus from the outside. So our immune system doesn't know that inside that virus there is a gene that is functional and is beneficial for the organism. So what our immune system is going to do when we inject into the circulation our viral vectors is that the immune system is going to attack it because it recognizes it as a virus. So this is the first issue with in vivo gene therapy. The second aspect is that we need efficacy. So as we said, we need to reach the right cell. So where is the genetic disease? I mean, the genetic diseases usually are systemic diseases, but there are, many times there are some districts or some organs that are more affected than others. So we need to restore the gene function inside the right cell type. Durability. How long is long term? So this is a very important concept. So with gene therapy, we have the issue of having one-time treatment that need to last lifelong. So this is very peculiar of gene therapy because if you think about it, there are no drugs that have a lifelong efficacy and there are no drug that needs to be lifelong, that needs to have a lifelong efficacy. Because we can take another pill. We can take another shot of vaccine if we need it. So this is very peculiar of gene therapy. And why this is a one-time treatment? Because did we say at the first point? Because our immune system will recognize the virus. So I think that the concept of that if you got measles, for example, when you were a kid, then you're not going to be infected again. So if you don't know what measles is, it's morbillo in Italian, for example. So you're not going to be infected twice. Why? Because your immune system will recognize very fast and rapidly the virus that already infected you in the past, and so it's going to prevent a second infection. And this is the same concept in gene therapy. So if we administer the viral vector once, we're not going to be able to administer it twice, because the immune system will prevent the efficacy of the second administration. Market. We don't like, as a scientist, to talk about market, but when we talk about rare diseases, we have to, because rare diseases are rare. So this means that there are very few patients that will benefit from the drug that we are developing. And also on the other hand, it's very difficult to have access to these patients. So if we need, for example, samples, blood samples from this patient, it's very difficult to get it because there are very few. And with very few, I mean that maybe for some genetic diseases, there are like few families in Italy, for example. So we are talking about a handful of patients, so it's very, very difficult. And also, for example, in low-income countries or in middle-income countries, these patients are not even diagnosed. So they don't even know what they have as in terms of disease. So it's very difficult to reach them. Money, other things that scientists don't like to talk about. These are very expensive drugs because if we compare a viral vector compared to a pill, this is a living drug. So it's very, very difficult, the manufacturing, the scalability of the process compared to a standard drug, let's say. And there are few patients, as we said. So if you develop a drug that is very costly, and then you have to administer it to like a handful of patients, and you can administer it only once, this, for capitalism, is not really a good investment. So this is an issue that needs to be taken into consideration also, how to price a gene therapy product that is a one-time drug only. So let's talk about our favorite viral vector. So in the lab, so there are a number of viruses that can be engineered to be used as a vector. So our favorite one, or at least my favorite one, are lentiviral vector. And we use this platform for gene therapy, and actually, I call it lentiviral vector, but maybe if there is some of you that studied virology at university knows that lentiviral vector correspond to HIV. So HIV, it's a pretty bad virus, as you may know, and causes an acquired immune deficiency syndrome that is lethal in humans. So we decided to use a vector that is very dangerous starting from a virus that is very dangerous for humans. So this means that we need to be very, very sure that we are not going to inject anything that resembles HIV anymore in the patient, because we need to be sure that it's safe, which is the first desire of a gene therapist, as we said. So this is how it looks like. Then I will show you also some real picture. This is just a drawing. So essentially we have proteins on the outside, and then we have a core inside that green cone, that two red S that are the genome of our viral vector. So that is the genome of HIV that we removed, so it's instruction manual, and we replaced with the gene of interest, okay, depending on the genetic disease. So on the outside, that blue mushroom that we have on the outside of the drawing, that are the proteins that give the specificity of the vector. What does this mean? So HIV is not able to infect all the cells of the body. So maybe you know it can infect mainly T cells that are lymphocytes, so cells of the immune system, or macrophages. These are the two main cell types that are both of them are cells of the immune system. But we said that maybe we want to correct the brain, or maybe we want to correct some genetic disease that is in the liver. So we cannot use the outside of the original virus, which is called envelope, what is on the outside. So we changed it, and this is actually what revolutionized the use of these vectors. And this is the work of Luigi Naldini, that is going to talk today at this Trieste Next conference. So if you can go listen to him, he's a very great scientist and speaker also. So, and this was actually the game changer in the use of HIV as a viral-derived vectors, because with this new envelope protein, we don't need to go into the details, but with this, we are able now to infect virtually all the cells of the body. So we can really use this vector for all to cure all the district that we need. This is a very technical design of the genome that we changed inside this virion. So also in this case, we don't need to go into the details. But here, so these three black rectangles, they are what we call the expression cassette. So this means that we say with that strength of instruction, this length of the instruction manual, we say to the cell, we tell to the cell how and when the cell, it has to read this sentence. Because the instruction manual that is inside every cell is not just a matter of what is written, but also when the cell has to read it. Because if we think about the fact that all the cells of our organism, they all share the exact same instruction manual. But if we think about a muscle cell compared to a neuron, they are doing pretty different things. And this is why is this? Because they read different part of this instruction manual. So we need to put inside our very small instruction manual and sentence, we need to have also the instruction on how and when to read this new sentence that we are putting back into the cell. Okay, so what did we do? How did we contribute to this? So we said that these lentiviral vectors, when we infuse them, they are recognized by the immune system. So we need to shield them in a way that the immune system is, let's say, we recognize it anyway because it's still a viral derived vector, so it's still deriving from a virus, but we need at least the first time that we administer it, we need it to be efficient. Okay, so it will be recognized, but we need the balance, so we cannot administer a second time, but we need the first injection to be efficacious, so we need it to be faster than the immune system in reaching the target cell. Okay, so what we did was to change how the viral vector looks from the outside. So this is an example on the bottom of the slide of how the viral vector gets produced. So essentially this thing in red, blue, and green, this is the viral vector. So this is the real, it's not the real colors, but this is a real image. So this is an electron microscopy image of a viral vector that is budding from the cell. So essentially what happened here, these are 3 steps. So as you can see, the protein core of the viral vector is encapsulated with the outer member of the cell that has infected. So this means that all the proteins that are present on the surface of the cell will be covering our viral vectors once it ends up the budding in the last part of the picture. So this means that we can change the protein composition of the producer cell that are much easier to engineer compared to the viral vector in order to enhance the biological properties of the lentiviral vector. So we're going to talk about very briefly only about these 2 molecules. So essentially the first one is CD47 and the other one is major histocompatibility complex 1. Why is this red protein important? Because the cell that we use to produce the vector are of human origin. So this means that most of the proteins that are on the outside of the viral vectors are of human origin. So our organism is not going to recognize them as foreign antigens, so foreign proteins, because they are of human origin. So most of my proteins are identical to yours proteins, because we are all humans. But there are some proteins that are different. Indeed, if you think about transplantation, if you need a liver transplant, it's very difficult to find a compatible donor. And why is this if we are all humans and our proteins are the same? And this is because there are proteins belonging to this family, so the major histocompatibility complex 1 and 2, that are very, very different among different persons. So my MHC1 will be very different from all of yours inside this room. And there are going to be, in the entire city, very few people that will have an MHC1 compatible with me. Okay? So what we decided to do is to remove this, because this can be an alloantigen, so can be recognized by the person as something foreign, so not of its own. So this can trigger an additional immune response against the vector or the cells that were corrected. So we remove it, first thing. So I'm putting also in the slide some references if some of you is curious and wants to go into the details of the scientific details of the publication. And this is an example also. This is an electron microscopy image. The black dots on the left that are all the MHC class 1 protein that we have on a standard lentiviral vector. And on the right, you can see that when we produce an MHC-free lentiviral vectors, we don't see that black dots anymore. The other protein that we were interested in is CD47. Why is this protein so important? Because we have in our organism different immune cells. But there is one specific type that are called macrophages that are called also scavenger cells. So essentially, their mission in life is to eat whatever they find around that don't look like yours. Okay, so if they find whatever things in your organism that is a bacteria, a virus, food, something that you are allergic to, okay, whatever, they will pick up it and present to other cells of the immune system. So what we can do is to enhance the amount of this CD47 that is present on the surface of our virion, because it is called also don't eat me signal. So essentially, its contact with macrophages block this process of capturing that is called phagocytosis. Okay, and for example, this is its density it's very, very high, for example, in our cells, in red blood cells, which is the reason why they can circulate so long in our body without getting captured by the macrophages. So we decided also to put a lot more of this, because of course, if we can reduce this uptake by macrophages, we can also reduce the presentation to the other cell of the immune system. And so we have a higher chance of getting into our target cell. And this is an example. So on the left, you have a standard vector. So you can see the whitish part of this round thing. It's the envelope, so the membrane part that I showed you. And the darker part inside is the protein core and the genome. On the right, you can see that that one has a lot of black dots, and that each one of these dots corresponds to a CD47 molecule. And also, if you look at the bar, those are 0.2 micron. So this means that these are 200 nanometers. So it's very, very small, this viral vector. I also put this video. I was very brave of including a video, because usually videos don't work, but today it worked, so we are happy with it. So please focus your attention. I know that the video is very nice, but focus your attention for a moment on the right. So I put a scheme just to elucidate what you see on the left. So in red, you see Kupffer cells. Kupffer cells are the macrophages of the liver, so these scavenger cells. In white, you see the blood vessel, which we call sinusoids in the liver. And then in green, fluorescent green, you have the lentiviral vector. So here, you will see when I will start again the video that you're not going to see the green. Why? Because it's too small, because we said it's 200 nanometers. But what you will see is that if you focus first your attention on the top left, you're going to see that the red cell become yellow. And as you learn at the elementary school, when you mix the red and the green, you have yellow. So this is why the cells become yellow. And why we can see it? Because it accumulates. So we have more than one virion. And so now we can see because we have a lot of signal inside the cell. So top left, focus your attention on top left. And then if you look at bottom left, you will see how it behaves, the CD47 high vector. So essentially, if you have a standard vector, most of it will end up inside macrophages. While if you look in the bottom left, you will see that the cells remain mainly red, and that only at later time points in the video, those that the timer hits minute after administration, you will see that only a few cells become yellow. So let's go back 1 second so that you can look at the video, if it starts again, but maybe not. Of course, because we couldn't be lucky twice. Maybe you can click, no, maybe not. On the play button, bottom left. Let's see if we can make it, otherwise you're going to receive the screen, the streaming, and go back just to see these few moments of video. Apparently not. Anyway, you have to believe me, or maybe you looked at it, the first play stamp that it played. So, what is today the landscape of in vivo gene therapy? In this slide, there are only the gene therapy that are already approved in the market. So we're not talking about preclinical research. So these are drugs that are really approved. So it means that a medical doctor can prescribe a gene therapy if you are born with a specific genetic disease. And as you can see, we have a few, okay? Not all of it are available everywhere because we have, as we said, issue in terms of market, so not all of them are available all over the world. But as you can see, we have different, genetic diseases that are written outside the squares. So hemophilia, Duchenne muscular dystrophy, spinal muscular atrophy, different diseases. We have different organs, so we can target the eye, the brain, the liver, spinal motor neurons, the skin, and the skeletal muscle. And we have then the name of the different gene therapy. So gene therapy is a reality today. So you can be administered a gene therapy if you're born with a specific genetic disease. And these are only in vivo gene therapy strategy, okay? So as we said, there are also ex vivo, but they're not present in this slide. So I would like to talk about the liver because it's my favorite organ. So as we discussed about my favorite viral vector, now we're going to talk about my favorite organ. So liver-directed gene therapy with viral-derived vectors has the potential to be curative with a one-time dosing, as we said. And by the administration of a viral vector inside the circulation, we can easily reach the liver because most of what is in our circulation then is filtered by the liver, which is one of the filter organs that we have together with the spleen, and so most of the blood pass through the liver. And so our vector, even if we inject it intravenously, most of it will end up in the liver and in the spleen. So we can administer intravenously and target the liver. There are approved gene therapy treatments for liver gene therapy, and these are gene therapies for hemophilia. Hemophilia is a coagulation disease, so meaning that people affected by hemophilia, we have hemophilia A and hemophilia B, so two types, and people affected by this genetic disease have a problem related to the coagulation of the blood. So this means that if we left untreated, if we leave untreated these patients, they are going to die because of bleeding, uncontrolled bleeding, okay? Of course, there are therapies that can be administered to this patient, a part of gene therapy, so we can replace these missing proteins by intravenous administration, so they are lacking two proteins, we are talking about it later, and we can administer by intravenous administration, but this is very costly and painful, and think about if you have to do 2 or 3 intravenous administration in a week. It's not an easy life. So there are approved gene therapy treatment for hemophilia, and these are based on a single intravenous administration of another vector. So not lentiviral vectors, as we were talking about before, but these are different vectors. So these are derived by these adeno-associated viruses that are human viruses that are not immunogenic. So this is a huge advantage compared of using an HIV-derived vectors, because HIV, as we said, it's a very dangerous virus. While if I use a virus that is not dangerous for humans, because it's not pathogenic, it's a smart choice, no? And you're going to think, well, why didn't you use these vectors then? We are going to talk about this. So in hemophilia, what they have shown, both in clinical trials, but also now in the market, because these are approved drugs, as we said, that we have multi-year therapeutic benefit in adult patients, especially in hemophilia B, which is one of the two types, while in hemophilia A, we have some issue in terms of durability. So how long will last the efficacy? So what we are observing in the people enrolled in this clinical trial is that now the efficacy of the therapy is decreasing over time. So we don't know how long it's going to last. Probably around 5 to 10 years. So these are the the perspective now. So what are the challenges for these vectors? So why aren't we using this? And we are also developing a strategy that can be complementary to these AAV vectors. First thing, they are non-pathogenic. So this means that there is a widespread pre-existing immunity to the parental virus. So most of us in this room, we have been infected already by these viruses during our life. So what does this mean? That our immune system, as we were saying before, is already trained to recognize these vectors. So if you are positive for antibodies against this vector, you are not eligible for the treatment of gene therapy because you are already immunized. So it's like you already received the dose, as we were saying before. So you cannot be treated. Second thing, HIV vectors, derived vectors, are integrating vectors. So this means that they take their own genome, so their instruction material, and they put it inside the genome of the cell that they infect. So when the cell then divide and give rise to daughter cell, each cell will have a copy of that corrected gene. If we use an AAV vector, they do not integrate. So this means that if I have one of these genome inside the cell and then this cell divide, only one of the two will have the corrected genome. And then these cells divide again, and then I will have 1 out of 4, 1 out of 60, and so on. So we have dilution of the genome, so of the gene, during, for example, in this case, liver growth, if I treat a pediatric patient. So I cannot treat a child. And also I have a packaging capacity, because I said that HIV-derived vectors are 200 nanometers. These are 30. So these are very small viruses. And so we have a packaging capacity. So the length of the sentence that we can put back inside that we can put inside these viruses is short. So it's not feasible for every sentence that we need to put back inside the genome. So in the last 20 minutes of this presentation, yes, still 20 minutes, sorry. In this presentation, we are going to talk about some application of what we have talked until now. So first of all, I want to talk about the difficult relationship between hemophilia and HIV, because we talk about HIV-derived viral vectors, and we have talked about hemophilia. I don't know if any of you know the story of hemophilia, but the relationship that it has with HIV, it's a very difficult relationship. So as we said, hemophilia is a coagulation disorder that is caused by the lack of coagulation factor 8, in the case of hemophilia A, or coagulation factor 9, so this is the name of the proteins, in hemophilia B. So here in this picture, this is Ryan, and this is a guy that was affected by hemophilia, and as you can see, he was born in the 70s, 1971, and he died in 1990, which is the year in which I was born, by the way. And during those years, this is what happened. So the only thing that a hemophilic patient can do in the 70s to survive was to get, first of all, blood transfusion when they were bleeding because of the disease, and also they received protein plasma concentrates. So the only way of treating them was to collect blood and plasma from healthy donors, like us, for example, concentrate this factor 8 and factor 9, based on if you have hemophilia A and hemophilia B, and then infuse them inside the patient. Why can we do this? Because as we said, most of our proteins are identical. Not all of them, but most, yes. So my factor 9 can be safely administered to another person. But what happened? It happened that most of these blood bags and plasma concentrates were infected by HIV. So during the spreading of the virus in the 70s and in the 80s and in the 90s, up to 60% of the US hemophilia patients get infected by HIV. And here on the right, you can see in these slides, these are the number of cases between the 1982 and 1989. And this is a graph that was published in the San Francisco Chronicles in the US, and you can see the amount of cases that were diagnosed. So these are cases of HIV in hemophilic population. So it's not just that you have a genetic disease that is life-threatening, but you also get infected with HIV. Not to talk about also other viruses, such as, for example, hepatitis C, which was another very life-threatening virus at that time. So it's a very difficult relationship. So if you think about it, so now we are proposing of using an HIV-derived vector to cure hemophilia. And hemophilia people were killed by this exact same virus. So pretty difficult relationship. So this is just a timeline of what we did in the lab. Also in this case, I'm not going into the details because we don't have time, but if you want, you can go and check the publication. So in 2012, our group, Alessio Cantori in particular, and that was in the lab at the time of Luigi Naldini, showed that we can use coagulation factor 9 encoding vectors, so an HIV lentiviral vector, as the one that we talked about today, that was where we put, in place of the HIV genome, we put the factor 9 coding sequence, so the correct sentence to produce again factor 9 that is missing in hemophilia B patients. So this vector was administered intravenously, so throughout the circulation, and reached the liver, and there corrected hepatocytes that are the main cell type of the liver, the parenchymal cells, that started again to produce factor 9. And this was done in hemophilia B mice. We then show efficacy also in hemophilia B dogs. And more recently, we showed that this vector can be safely administered and used in non-human primates, which is a necessary step before testing in humans, of course, because as we said, you need to prove safety first. So you need to be sure that this is not going to cause an immune reaction that can be fatal sometimes. And this happened in the past with other type of vectors, not with HIV-derived vectors, but other types of vector that have been administered in person. And actually that guy died because of a very strong immune reaction against the viral particle itself. This is why I'm stressing this aspect of safety, because our immune system is very powerful and can kill you if it gets activated too much. So it's very important to be sure about safety and to do all the preclinical work also in non-human primates to be sure that your treatment is going to be, yes, efficacious, but safe first. So we think that lentiviral vectors can be a complementary strategy to adeno-associated viral vectors, which are the vectors that are already in the clinic, as we said. So this is just a recap. So we showed stable gene transfer in adult immune-competent mice and dogs, a dose-dependent reconstitution of the clotting factor activity. So if I administer a higher dose of vector, I obtain higher amount of factor 9 in the circulation. We have very mild acute toxicity and efficient gene transfer even in non-human primates. And so this is just one piece of data. So you can see on the left the level of expression of factor 9, in the case, which is the curative gene in the case of hemophilia B, and on the right of factor 8, which is the curative gene in case of hemophilia A. And you can see on the x-axis two different doses of the two vectors, and you can see that we can reach even supraphysiological level because the blue dashed lines are the normal range. So this vector can be actually efficacious in providing an amount of factor that is curative, because it's not just that you have to put the gene back, but you also need a certain production of the protein that you need to get what we call a normal range of activity. And we are now working toward clinical evaluation, so a possible clinical trial in humans. Lastly, in the last 10 minutes, I would like to talk about new avenues for in vivo gene therapy. So we have talked about mainly, so as a target, I showed you a picture where all the targets were solid organs. Okay, so I showed you the brain, the eye, the liver, the muscle, so solid organs. So this is the picture that I showed you before. Actually, the new avenues for in vivo gene therapy are even cells of the immune system and hematopoietic stem cells. Why? Because as I said, typically these cells as a target were treated with approaches of ex vivo gene therapy. because it's very easy to collect these cells. I collect it from the blood or from the bones, I put them in culture, I put the vector, and then I infuse back the cells. Easy. This is easy only if you are in a lab. But let's say that you are in Indonesia, in an island, and then you need to go do a transplant, you need someone that is able to do a transplant, you need to stay in the hospital because you get chemo, because you need to make space in your own bone marrow then if you have to do a transplant, even if they are your own cells. But you need to make space for them to engraft. So you need a chemo. So you have to do a chemo, you have to stay in the hospital, you need to have a facility that is able to culture your cell. You need to have a viral vector shipped there, and someone that is able to put it on your cells, preserve the stem cell function before you infuse it back. And for every patient, you will end up with a different drug product, because the drug product in that case is not the lentiviral vector, because what you are infusing in the patients are the corrected cells. So my cells will be different from yours, so each patient will have a different drug product that needs to be characterized, and this is very complicated and costly. So this is why the field is trying to move to in vivo gene therapy approaches even for this type of diseases. So for example, T-cell that I wrote on the right, that is a very interesting paper if you want to read it. It's a very recent publication a few months ago, and this is a clinical trial in humans. So they modified the lentiviral vector that I showed you before, and use the same, our technology that I showed you, so vector without the MHC1 with CD47I, that are CD47I, and they, in that case, they generated directly in vivo CAR T cells that are reprogrammed T cells that are able to attack, in this case, the tumor, which is, in this case, multiple myeloma. And this is already clinical trials in human, so very interesting. On the other hand, which is still at the preclinical stage, people is working to do this in hematopoietic stem cell, because if you can target hematopoietic stem cell, then you can correct all the daughter cell of the hematopoietic system. And this is actually what I'm working on now. So as a project leader now, I started this new project on targeting in vivo hematopoietic stem cells. And so this is exactly what we said, no? That if we do an ex vivo hematopoietic stem cell gene therapy, we need to manipulate the cell. There are the risks associated with the chemo and the transplantation. You need to be immune compromised for a period. You are at risk of infection. You need a specialized center, so it's very complicated. On the other hand, on paper, an in vivo gene therapy strategy would be much easier, because you just need to ship the vector batch, and then you have just an intravenous administration, which is much easier compared to a transplant. However, there are a lot of hurdles also here and challenges, because otherwise we would have done it already. And first problem, low efficiency. What did I say before? That if I administer an antiviral vector intravenously, most of it will end up in the liver and the spleen, because these are our filter organs, and this is a virus. So our filter organs will trap most of the vector. So we have very few vector particles that are available to reach the hematopoietic cells that are in their niches. We lack specificity of targeting. If I collect the cell and I put them in culture, I only put the vector on those cells. If I put the vector in the circulation, as we said, we reach the liver, the spleen, other targets on top of the hematopoietic cells. And we need to target the cells in their niche. So ex vivo gene therapy in hematopoietic and progenitor stem cells, so we didn't talk about this until now, has proven over the last decade to be curative for a number of genetic diseases. Curative. And also in this case, there are drugs that are already approved in the market. So if you are born with a specific genetic disease, your medical doctor can prescribe a gene therapy, an ex vivo gene therapy. But there are challenges. In some conditions, the number of collected cells could be limiting. If I have a problem in my hematopoietic system, it's possible that I don't have enough stem cell to be collected, engineered, and then reinfused back. Cells need to be kept in culture and manipulated. It's possible that during the culture they lose part of their stem cell capacity because they're not in their environment. The transplantation needs to be performed in specialized center, as we said. In vivo gene therapy is also challenging, but can be complementary to ex vivo gene therapy. So we need to target the cells in their niches. So which are the niches of the hematopoietic stem cell? In adults, usually they stay in the bone marrow, so inside the bones. But they can also be in the circulation in specific time frame of our life, or, for example, if we get a mobilization protocol. So there are drugs that can be administered to mobilize the cells from the bone marrow and force them to go into the circulation. Or they can be in special niches. For example, during the neonatal or prenatal life, during the fetal life, our hematopoiesis takes place in the liver and in the spleen and not in the bones. So it moves afterwards. So we need to target the cells where they are. It may be advantageous when there is a selective advantage. What does this mean? If I have a problem in my hematopoietic system and I correct some of the cells, it's possible that these cells have a survival advantage over the non-corrected ones. So this means that they proliferate, and then even if I correct a few of them, over time they are going to duplicate and expand because they are corrected, and so they have maybe a proliferation or a survival advance compared to the non-corrected ones. And why this is important? Because, as we said, we have low efficiency because most of the vector goes inside the liver. And the other thing is that, as we said, we will transfer gene to other organs. And this may be a disadvantage in some cases, but also an advantage in some other cases. Because in most of the cases, genetic diseases are systemic diseases, okay? So it's true that there is one specific district that is causing the death of the patient, but most of the time also the other districts are affected. And if we are administering a one-time cure with one-time dosing, and then we make the person survive, but then it has a lot of other problems in all the other organs, it's going to be a problem. So we need to be sure that we correct most of the tissue that are involved in the disease. But is it possible to target HSPC, so hematopoietic stem and progenitor cells, in vivo by a systemic administration of lentiviral vectors? So what we did, we started by studying the biology because we are scientists and we are curious. So we like to cure diseases, but we also like to learn new things. So this is a very simple graph. So on the left, you have the number of hematopoietic stem cell, HSC, throughout the life of a mouse. So we have newborn, so this is 0, 1, 2 day, 1 week, 2 week old. 2 week old means around weaning, so 5, 6 months in humans. 8 week old, which is an adult. Easy. 6 weeks, 30 years of life. And then on the left, you see the number of cells in the bone marrow. On the right, you see the number of cells in the circulation at the same ages. And essentially what we discover is that we have a high number of circulating HSC, so hematopoietic stem cells in newborn, that decrease over time, on the graph on the right, and you can see that in parallel we have an increase in the number of cells in the bone marrow. And this is because, as I said, in the fetal life we have the hematopoiesis taking place in the liver and the spleen. And then the cells during the very early, so we are talking about few days, postnatally, these cells migrate from the liver and the spleen to the bone marrow. And so we have a decrease of the number of cells in the circulation and an increase of the number of cells in the bone marrow. So we perform a very stupid experiment. So what happens if we administer a lentiviral vector, so an HIV-derived vector, the one that we have talked about in this presentation, in this case expressing what we call a reporter gene. So this is just a green fluorescent protein, GFP. So it's nothing curative, it's just a protein that can be easily visualized because it's green and it's fluorescent. So we administer it intravenously in a newborn mouse, and then we follow what happens in the blood of this mouse for 1 year, which is a very long time for a mouse, because a mouse lives more or less 1.5 years, maximum 2 years. So it's going to be almost lifelong for a human being. And this is what happened. If we look at GFP positive cells, so cells that are green in the circulation, you can see that in all the major, let's say, cell type that we have in the circulation, which are myeloid cells and lymphocytes, so T and B cell, you can see that we have very few, just 1% of cells that are green, but this is stable throughout the entire life of the mouse. And this means that we are not targeting already differentiated cells, Because you can think, no, I'm administering the vector in the circulation. In the circulation, there are already T cell and B cell and myeloid cells. I'm putting the gene, the green gene, inside these cells, and of course I see them green. But if this was the case, then I should have seen a decrease over time, because these cells has a very fast turnover, because we have the hematopoietic stem cell that are in the bone marrow that generate the cells of the blood very frequently. So every week we change our blood cells. Then we have some few cells that are long-lasting. For example, T cells stays in the circulation longer compared to the others. But myeloid cells, for example, they have a very fast turnover even in humans. So this means that if we see this GFP staying for the entire life of the mouse, this means that we have targeted actual stem cell and not differentiated cells. So this is important in terms of durability, because we want our, as we said, we want our strategy to last for the entire life of the patient. So we need to target actual stem cell. So we applied this to different genetic diseases. We are not going to talk about them. I just put them here to show that it can be applied to more than one genetic disease. We are just touching briefly on ADA SCID, because we are aficionados of this disease. And I'm going to talk just in one slide about the long journey from the bench to the bedside of Stream-Velis. So let's start from the disease. The disease is adenosine deaminase deficiency. And essentially, the lack of this enzyme, so a lack of this protein, is causing a severe combined immune deficiency. And maybe you have already heard about this. For example, there is one of the Grey's Anatomy case was about this disease, and there is also a movie about this disease. And these kids became sadly famous as bubble kids, because the only way they had to survive was to live inside this bubble that actually prevented their contact with all the pathogen that the bacteria, the viruses, whatever that lives in the environment. And as you can see in this picture, there is this doctor that is visiting the patient through these gloves that are separated from the outside to the inside of this bubble. And this is the long journey that actually Stream-Velis did. Stream-Velis, it's an ex vivo gene therapy for ADA SCID that was developed at the San Raffaele Teleton Institute of Gene Therapy by Alessandro Aiuti, Professor Alessandro Aiuti, Maria Grazia Roncarolo, and Claudio Bordignon and the team. And it was 2002. I was just finishing my primary school. I didn't know that I had to become a scientist in my life yet at that time. In 2016, Stream-Velis was approved as a drug. So if you are born with ADA SCID, you can be administered Stream-Velis and prescribed by your doctor. And this was only possible by the alliance of the San Raffaele Teleton Institute of Gene Therapy and GSK, which is a big pharma that had the vision at that time to invest and put a lot of money in the development and the clinical trial of this disease. And then at a certain point, this was passed through Orchard Therapeutics, which is another pharma. Unfortunately, in 2022, Orchard Therapeutics discontinued the investments. And this is because rare diseases are rare, as we said. And this is a huge cost to maintain the market approval of these drugs. And then there are very few patients that you can be treated, because these are few patients. Some of them don't even get diagnosed. And maybe they need to travel from Asia to Italy to get or to Europe to get the treatment. And then Fondazione Teleton decided to acquire the Stream-Velis marketing authorization. And this is the only case in history where a non-profit entity has acquired the marketing authorization of a drug. And this is very important, because Fondazione Teleton didn't accept that we have a cure for a genetic disease that then at a certain point is not accessible anymore just for money reason. But this is not because of the big pharma. This is because this is the world that we built. So we have this type of economy. So we have to fit in. so Fondazione Teleton decided as a non-profit to get back the marketing authorization. So about this, we don't have time to go through the details of the science. I just want to show you that this in vivo gene therapy strategy, and we did this in collaboration with Alessandro Aiuti because, of course, we are in the same institute and it was an obvious, let's say, and fruitful collaboration together. So we tried our in vivo gene therapy strategy in this disease model in mice, and this is because, as we said, the vector goes inside the liver, you remember, and the liver can act as a protein factory, so can produce the enzyme and can help the detoxification of the entire organism, even if we need to target also the hematopoietic stem cell. But in this case, the targeting of the liver is beneficial because ADHD is a systemic disease, as we said. And here, just look at if you look at the gray dots on the left, this is the survival of the mice that left untreated. So if you don't treat them, in 2 weeks they die. Of course, we don't have bubbles for the mice where we can put them. On the other hand, if you look at the purple dot, focus your attention only on the purple, those are the mice that have been treated in vivo as newborn with the lentiviral vector encoding for ADA, so the enzyme that is missing. And as you can see, around 70% of them survive long-term. 20 weeks is long-term for a mouse. So this means that we can cure this disease in most of the treated mice, even with an in vivo gene therapy strategy, which is very important. This is just to tell you, if you look on the right, that of course all the data that I showed you were related to mouse models. Is this window in the postnatal period, does this exist also in humans? So we did this also in collaboration with Dr. Serena Scala and Alessandro Aiuti. If you look on the right, this is the same of what I showed you before. These are HSC, so hematopoietic stem cell, number in the circulation, so this is peripheral blood of humans. So as you can see, we have a huge amount in neonates of hematopoietic stem cell in the circulation. So this means that this trafficking from the liver and the spleen toward the bone marrow happens also in humans in the first days after life. So it's possible that we can take advantage of this window of opportunity even in humans, of course, in the future. We are talking only about preclinical work. So to go back to the wish list of a gene therapist, as we said, we need safety, we need efficacy, we need durability, which is key in gene therapy strategies because we have the opportunity of treat the patient only once. We have to face issues with the market and we have to face issue with the money because of the rarity of the diseases and the economic models that needs still to be to evolve for this type of diseases that up to now, we didn't have this problem up to now because there was no cure for genetic disease. There was just treatments. And treatments you can administer more than once. So it's a different paradigm, completely different, that the entire world needs to face and adjust to. So my take-home message for today is that there is nothing so bad that is not good for something. So you need to keep this in mind for your life, for your future studies, whatever you're going to study. Be brave, put videos in your presentation, and trust that even if something looks very bad, most likely you have to have the eye to find something good in it, and usually the most powerful ideas come from this type of observation. I also want to shout out to this international PhD training program, which is Rare Find, and since I know you are university students, so this is a new program that we are about to launch. The call opens the 1st of October, and you can scan the code or go to the website for more information. This is a program created by Fondazione Teleton and the use of Pavia, and the lab are going to be in the SR-TIGET in Milan, where I'm from, and also TIGEM, which is the other institute of Fondazione Teleton, which is in Pozzuoli near Naples. Much more beautiful than the TIGET because it's by the sea, But also in TIGET we have nice people, so also you can consider coming to TIGET. With this, I would like to finishing. I already mentioned some of these people. I know I don't have time to go through all the names, but this is just to tell you that science takes a village. I want to say thank you to Luigi Naldini and Alessio Cantore, which are my two main mentors. Remember, mentors are essential throughout your growth, so choose a right mentor. And if a mentor is not it's not fitting you, change mentors. It's not that you have to change the mentor. And also, I want to tell you this last proverb. Yes, I like proverb. If you want to go fast, go alone. But if you want to go far, go together. And this is a key in science, and not only in science. Of course, I'm a little bit biased toward science, but this is true also for all the other areas of study. So remember that if you want to go far, you have to go together. Thank you. So thank you all. I've seen that most of you remained awake for the entire duration of the talk, so thank you very much. So I don't know if you have any question. Feel free to ask. We have, I think, still 10 minutes. Don't be shy if you have a question. I'm sure I wasn't clear for everything that I told, so, or if you have any curiosity or whatever, you can ask. And if I don't know how to answer, I will invent something, so don't worry. You can ask whatever you want. And also, if there is something that comes to your mind later, you can drop me an email and contact me later. Yes, sure. If you have additional questions while I go back to the email. Yes?
First of all, congratulations for a amazing lecture.
Thank you.
I have a question about this slide. So here you show that the GFP is like expressed at like 1% of the cells, and thereby you show that it's like targeting the stem cells. Do you have like a specific envelope protein that targets specifically hematopoietic stem cells on that vector, and also the promoter? Is that like a constitutive promoter that is like?
Yeah. So thank you for the question. Yes, so the PGK promoter is an ubiquitous promoter, so it's expressed everywhere. And very smart question. So this vector is just the wild type vector that I showed you, so it's pantropic. So this means that this 1% is most likely so low because the vectors enters very efficiently in the other organs. So we lose most of the vector inside the liver and the spleen, and 1% of it enters into the hematopoietic stem cells. Actually, one of the future perspective of this work is that we are working on engineering the envelope of the vector in order to retarget it and detargeting it from the liver and trying to retarget against the hematopoietic stem cell. But this is more related to for the efficacy, no? Because if you are a vector that is retargeted specifically toward your cell of interest, most likely you are going to increase the number of particles that are available for those cell type and increase efficiency. I didn't show you, but we have means of to increase this 1%. So if you want to go into the details, there is a Nature paper on it. And we are able, let's say, to reach between 15 to 20%. So with this vector, this is the maximum that we were able to reach. Thank you.
I have a question about the survival advantage of modified cells. I was wondering, are there ways to influence that advantage with the vector, or is it just random and you can choose which vector shows you the best advantage to the modified cells?
Okay, thank you. Also in this case, very interesting question. So usually what gives the selective advantage to the cell is the fact that they are cured. So for example, there is a disease, I didn't have time today to talk about it, which is Fanconi anemia, that essentially is I'm not able to go back anymore, is a genetic disease in which it's the stem cell, so the hematopoietic stem cell itself, they die. So once you put back the gene, this cell starts to survive. And since the bone marrow niche is emptying because the other cells are dying, these cells are prompt to proliferate to recover the niche. And so in that case, you have a selective advantage, which is due to the biological properties of the corrected cells and not by the vector. There are other cases in which, for example, you can add an additional piece to your cassette inside the vector, and you give, for example, an advantage to a chemotherapy. And then you treat the mouse or the patient or whatever with the chemotherapy, and then you kill all the other cells except the one that you corrected. And so, once again, that cells then expand because, so there are two types, let's say, of selective advantage. One that you can install inside the cell, and another one that is given by the fact, just by the fact that the cells are corrected. And so now, and it would be the same even with the transplant in that case. Thank you. Other question? Do we have time for one question? Last question.
Okay, let's focus on the liver. That's the closest thing to you, right?
Yeah.
So the drug used for HBV,
Mm-hmm.
I mean, does it have like a tropism for liver, and how it is like directed just to the liver, and maybe some comment on the side effects of that thing. Actually, is there a possibility to cause some other aberrations?
Yes. Other very important aspect that I didn't have the time to cover today. So regarding the liver, so the reason why most of what you administer intravenously goes into the liver is because of the liver architecture. So the liver has a lot of blood vessels that are called sinusoids that are meant to filter the blood. So the caliber of this vessel decreases very much, and there are a lot of holes inside these sinusoids that are called fenestre, and this is the reason why most of the things that are administered through the circulation ends up in the liver. Every drug, in general, is filtered by, most of our drugs are processed by the liver, even the small molecules that we received. In the case of AAV vectors, the specificity of the vector is given by what we call the capsid, because in that case you don't have the envelope that you have with lentiviral vectors. So there are different families of these viruses, and according to the vector that you use, you have a little bit different tropism. But all of them goes inside of the liver because of this filtering capacity of the liver. Then on top, you might have additional other tropism. So for example, the muscle, the brain, but most of what you administer will end up in the liver, unless you detarget the liver somehow. But this is not the case for what is now in the clinic. Regarding the adverse effect, so there are, I would mention two, which are the main one. So regarding lentiviral vectors, these are integrating vectors, as I said. So they integrate randomly into the genome. So this means that they don't have a specific region where they get integrated. So what can happen is that they can disrupt some other sentence in our instruction manual. Usually this does not happen with these vectors, but if you put some specific let's say sequences inside your cassette that, of course, you put them on purpose to study tumorigenicity, then you can cause what is called insertional mutagenesis. So a mutagenesis that is caused by the fact that you are inserting something in the genome and you are interrupting another gene, or you are interfering with other genes. And in that case, you can have an hepatocellular carcinoma. In mice, what we have shown over the years is that this does not happen. So it's a theoretical risk, but with this specific design of lentiviral vector, it doesn't happen. While if you change on purpose the design of your vector, then you can trigger this type of effect. But it's something that, of course, needs to be taken into consideration when assessing the risk-benefit of the therapy. With AAV vectors, so not in the context of liver gene therapy, But with other gene therapies where you need to go very high with the doses, because people was confident in increasing the doses because it's a non-pathogenic virus. But actually what happened is that some patient died because of these very high doses, because they had, let's say, blood-related consequences, so thrombocytopenia and disseminated coagulation, and multi-organ failure because of the very high dose. So essentially, also if the vector is non-pathogenic, you need to be careful in all the toxicity studies in the non-human primate setting. So thank you all. We finished our time, so if you have any other questions, just drop me an email. Thank you.